Um, okay, so my name's James. I come from the University of York in the Department of Biology. I am new to this whole idea of microbial communities. My background is in cancer research and DNA replication, rather bizarrely. But I'm going to talk to you about methanogenic communities and how I've been interested in them for the last few years. Okay, and I thought I'd start with this because this is the best picture I have. So this is from Christmas Day 2012, when I was down on Cygney Island, which is just inside the Antarctic Circle, collecting some seawater samples. And this is Coronation Island, which most of the time you can't see. This was a very unusual picture because it was sunny and clear. Okay, so like I said, I'm interested in methanogens. So I'm going to talk a little bit about what methanogens are, which perhaps is not something this audience particularly needs. So I guess the first thing that I have in my previous life always made a big fuss about is that methanogens are not bacteria. They are microorganisms that look like bacteria, but actually biologically they're quite different. And that's really where my interest originally stemmed from. I was interested in DNA replication. The replication machinery in this whole group of organisms called archaea is more similar to the replication machinery found in eukaryotes. And so I originally came to this field using archaeal organisms as a model for human DNA replication, simplified model for human DNA replication. Okay, so methanogens probably are not the best organisms to use as a model for DNA replication in humans, just because they're a real pain in the neck to grow. But we'll talk about that in a minute. So there's a whole bunch of these organisms that grow in a whole range of niches. They've been isolated from the Antarctic all the way through to organisms that grow above 100 degrees. All sorts of different morphologies, and the thing they have in common is this unique biochemical pathway which allows them to produce methane in order to generate energy for their cells. So methane is a waste product for these organisms. And they can do it a number of ways. Classically, by reducing carbon dioxide using hydrogen. Actually, most methanogens ferment acetate, but there are also a nice group of organisms that do slightly different things with methanol and methylamines in order to generate methane as well. And so my lab's big interest has been in using this organism, Methanogenic maripaludis. This is actually a marine organism. It's a very strict anaerobe, and that's one of the big challenges in growing methanogens on their own. Otherwise, this organism is quite a lot like E. coli in terms of its flexibility. It grows around 37 degrees at optimum temperature. It has a sequenced genome. It can be cultured in the lab, and there are actually well-established genetic systems for this organism. So you pretty much can do everything that you can do in E. coli and Methanogenic maripaludis. You can knock genes out, you can put them in, you can overexpress them. But because methanogens in general are a real pain to grow, very few people use these organisms. So until very recently, my group was the only group in the UK that grew methanogens routinely on their own. And we're still the only group outside of North America that does genetics in these organisms. And there are probably only half a dozen groups worldwide that routinely do genetics in maripaludis. There are a couple of other model methanogens that you can also do genetics in. They're even more difficult to grow than maripaludis. Okay, so this is what I hadn't realized when I embarked on growing methanogens, that you need quite a lot of infrastructure to deal with them. So we have anaerobic chambers. Methanogens are very strict anaerobes, so that means that they only grow in less than 10 parts per million oxygen. Now, in an environmental sample, that's probably easier to maintain because you have other organisms that are scavenging the oxygen. When you're trying to grow them in pure culture, it's a nightmare because it turns out oxygen sticks to everything at that kind of 10 part per million level. So all our plastics, all our metal, everything that we use has to go inside one of these anaerobic chambers at least a few days in advance, because otherwise the oxygen that's stuck to the Petri dishes kills the organisms. The other big challenge is, so maripaludis grows on carbon dioxide and hydrogen. you can buy pre-mixed hydrogen CO2 cylinders, but they're expensive. And so for cheapness, we have a hydrogen cylinder and a carbon dioxide cylinder, and we mix the gases ourselves. But the hydrogen that you can buy cheaply is only 99.99% pure hydrogen, which leaves you with more than 10 parts per million oxygen in the gas. And so we have to scrub the gases. That's what these things are. These are basically homemade gas scrubbers, piece of copper tubing with copper inside. You heat them up to about 180 degrees centigrade. You put your gas mixture over the top. Any oxygen that's present reacts with the catalyst to make water. And then you get pure hydrogen coming out the other end. So needless to say, the first time we turned these on, not many people stood around to check that we got it right. But having said that, once you've got your gases sorted out, we can grow these organisms in these things that we call bombs on plates. So we can isolate individual colonies, and we can grow them up to, fermenter size. Our fermenter is about a 2.5 litre working volume. So we can grow from single colonies up to that kind of scale fairly easily. Okay, so as a community audience, I guess you know all this. Methanogens are ubiquitous in the environment. They were first described by Alessandro Volta, who wrote a letter to his priest for some inexplicable reason in the 1700s, talking about combustible air. So Volta, when he wasn't busy playing with batteries, used to take his servants out into the North Italian countryside and go around setting fire to marsh gas, which is really the vision of methane production in the environment. So, of course, when you hear about this Volta experiment and you work on methanogens, it's one of those things you have to do. So the University of York has an artificial lake. It's the largest rubber-lined lake in Europe, apparently. Lots of things drop into it. And the students are strongly discouraged from swimming in it because it's not very nice. So I took a willing postdoc out into the lake, and we wandered around for a little bit trying to collect bubbles to really demonstrate that methanogens can make methane in the environment, which I didn't think would possibly work. So in about 15 minutes, we managed to fill a big funnel and very nearly set fire to the trees. It's absolutely incredible, and I was really pleased that this worked. We've tried it a couple of times since. It works all the time. So there's huge amounts of methane being produced in the environment from the breakdown of organic matter, not just by methanogens, but by a whole consortium of microorganisms trying to get their slice of the carbon, basically, that's being dropped into these kinds of situations. The figures on this are not brilliant. This is from a publication from quite a few years ago, which suggests that about 1 billion tons of methane are generated per year by methanogens in the environment. Most of that, fortunately, does not go into the atmosphere, because if it did, we'd be an awful lot warmer than we actually are. Figures are that methane has 25 times the global warming potential of carbon dioxide. Anthropogenic activity only puts about 8 billion tons of carbon dioxide into the atmosphere. So if all of this went in, that would be about 3 times as much global warming potential as we generate from CO2. Most of the methane is captured by other organisms and resequestered. So only a little bit escapes. But most of the methane that is emitted is through methanogenic activity. Methanogens live in termites. They live in a lot of farm animals, such as cows. Obviously, they can grow in paddy fields where the soil underneath the water is anoxic, but they're also found in landfill sites, sewage treatment, animal waste. So most of the methane produced is from methanogens. Okay. So apart from all those environmental niches, you find methanogens with most microbial communities, anaerobic.
Can you say something about where the methane goes then? Right, so it doesn't go to the atmosphere, so...
Okay, so there's a whole other group of organisms called methylotrophs, which will take that methane and convert it into methanol, and then the methanol gets fixed into longer molecules again.
Just one further question. You mentioned natural gas as a source of methane. What is it a source?
Right. Well, yes, but not so. Okay, so these other figures are the other emissions that we get. So you get methane from mining, basically, as you know. Most of the time it's flared these days, but there is a certain amount of escape. And in fact, people are starting to find, even in spent mines now, they're injecting methanogens to deal with the carbon dioxide and hydrogen there and generating methane that's then being tapped off again. One of the other big issues is landfill sites. So the way I explain this to my students is, you know, if you're not eating burgers, which are producing lots of methane, in fact, cow emissions are responsible for nearly a third of the UK's methane emissions. You're eating sushi, and when you're not eating, and both these things generate a lot of waste that goes into landfill sites that also generates methane. Okay, so methanogens are also found in other anaerobic environments. So the human gut is one of them. The numbers on human gut methanogens are horrible, as in they're very, very vague. The typical way people have measured the presence of methanogens in human guts is by breath methane, which is not the best way of measuring whether there are methanogens there. The other thing that people have realized quite recently is a lot of these organisms are extremely difficult to break open. And so if you don't use the right kind of protocols to analyze stool samples, you don't see the organisms because you're not getting DNA out of the cells. But something that I think is very interesting is, so the organism that's been most commonly identified from the human gut is this Methanobrevibacter smithii. It's had its genome sequenced. You can grow it in the lab, and when you do it, it looks like this. When you isolate it from a community growing in a human gut, it looks like this. So all this exopolysaccharide around the organism just isn't seen when you grow it in pure culture. And I guess that's a really important thing to think about, for me at least, in communities, that these organisms behave completely different when they're grown on their own to how they behave when they're actually living in a complex community. And there's been some really nice papers recently, or relatively recently, by Jeff Gordon's group looking at the co-culturing of Methanobrevibacter smithii and Bacteroides theta iota micron, showing in mice at least that you can see increased weight gain when you have both these organisms growing in your gut compared to either of them on their own. So there's all sorts of, you know, I haven't managed to work it into a grant application yet, but there's this potential that methanogens might make you fat. The other thing that has been a big problem actually is because methanogens belong to archaea and there are no known pathogens in the domain archaea, actually it's very difficult to sell working on methanogens because they don't make people sick. So that's been another reason that we've kind of gone in this direction of looking at communities that have got methanogens in that might be useful rather than harmful. Okay, so methanogens are very abundant inside these organisms. So the typical figure quoted is that a cow on a poor diet, I'm not quite sure what a poor diet is for a cow, but I think it involves quite a lot of grass. A cow can produce about 200 litres of methane a day from belching, but those belches are all methanogen driven. So it's the methanogens in the cow gut that's generating all this methane. And so people are doing a lot of experiments, particularly in Australia, to try and reduce the methanogen load in cows and sheep so that they don't emit as much methane. There are ways of taking advantage of these communities, and that process is called anaerobic digestion. It involves taking organic waste. In this case, this is just farm waste from a dairy farm, putting it into a large container, stirring it a little bit, and then taking the gas that comes off the top. That gas is a mixture of methane and carbon dioxide, and it's typically fed into a combined heat and power system. So you basically burn the methane, you generate electricity, which can be fed into the grid, and you can use the low-grade heat, which comes out around 80 degrees to power other systems. In the US, the EPA have this AgStar program, and they've been collecting data since 2000 on anaerobic digesters across the states. And it's been fascinating to watch this in the last few years. So these are their latest figures. These are the greenhouse gas emission reductions by taking essentially the slurry that cows produce and digesting it rather than just letting it lie on the ground. For 2013, they reckon they saved 2.53 million metric tons of carbon dioxide equivalent from digesting cow slurry and using that. And if you turn that into the amount of electricity or energy that you're generating, you're looking at about 840 million kilowatt hours. And this is over a very few relative sites in the state. So they are tracking what's being generated out of about 250 anaerobic digesters across the states. So not a huge number, although some of them are very large. So I can never work out exactly how much 800, well, and you can see the number keeps going up. I reckon that's enough to run all of York and Cambridge, all the houses in York and Cambridge for about 2 years, being generated by cow waste. Okay, so in terms of community, we know the community structure that's involved in breaking down organic waste is complicated. What you're doing is basically taking all the molecules that most other organisms have spent their time building, proteins, polysaccharides, and lipids, and hydrolyzing them down into monomers and oligomers, and then fermenting those into intermediates, which ultimately feed into methanogenesis, either through acetogenesis or through gas production. And so this is being used a lot. We got involved with very early in the time that I was interested in this. We got to know about an anaerobic digester that was taking tomato waste and trying to make gas from that. So this is one of the UK tomato growers. They produce about 1/3 of one particular sort of tomato for one supermarket in the UK. And their standard generation is about 10 million tomatoes a week during the growing season. So they have a lot of plants that are grown hydroponically. They produce a lot of plant waste, and it was costing them about £2,000 a week to landfill their leaves from their tomato plants. So instead they built this anaerobic digestion system. They mash up the leaves and put it in at one end. This is actually a multi-tank system, which is relatively unusual in anaerobic digestion terms. The ground up material is, given to microbes, in this case again cow slurry basically, and then move from tank to tank, and you get gas out of the far end. Okay, so we took some samples from those tanks and just had a look to see microbially if we could see anything interesting. So tank 1 is at the beginning of the system, tank 4 is at the end. When we isolated DNA and then did just a 16S PCR specifically for archaeal organisms, what we can see is all the archaea appear towards the end of the system. They're going to be mainly methanogens, we speculate. If we look at bacteria, it's the other way around. There's nothing being managed here, so the organisms are spontaneously forming a gradient down these tanks. And this is just doing DGGE to look at a similar sort of thing to show that there's more than one organism and that they are different organisms in those different systems. Right, so we were hoping to use that model system to understand a lot of what was going on, but I guess, as is not unusual in these kinds of operations, the person involved in running the system became unreliable, or let's say did not have exactly the same ideas about what constituted a useful system to our academic as others. So they were constantly changing it, which made it very difficult to draw any useful conclusions from it. So my group's now started collaborating with another anaerobic digestion company, this company Clearflow, who builds systems specifically to go on food production sites. The nice thing about food production sites from our point of view is that generally most sites only produce one type of food, and so the waste streams that they're dealing with are fairly consistent, and they tend not to have any exogenous microbes in them because clearly you don't want to be producing food that's contaminated with microorganisms, generally frowned upon. So this is quite a nice model system from us. They have a small pilot scale system, which is 8,000 litres, which goes in a container that can go on the back of a lorry and be delivered to different sites. And so they can basically go to a site, plug it into the waste stream that's coming off that site, which is normally wastewater, and demonstrate that their process works before the company invests several £million in having something built at full scale. So this company has been sponsoring a PhD student with me, and we've tried to replicate their 8,000 litre system in the lab, which is shown here. So we are using saucepans to try and do exactly the same thing. So these have a working volume of about 30 litres. We have 4 saucepans in the lab, and the idea is to see how reproducible this system is, how robust it is to getting the same organisms out if we feed them the same thing every time, and what happens over time. The nice thing is, of course, that we can sample from these systems. So this is basically the schematic. We can inoculate our system with a mixture of microorganisms. At the moment, we're using an anaerobic digester that is run by Yorkshire Water and is actually, fortunately, sited about 3 miles down the road from where the university is. And that seems to work fairly consistently. It takes all the wastewater from York and processes it. And they are now at the point where they're producing so much gas, they're having to vent some of it because their CHP plant isn't big enough for the amount of gas that they're producing. We feed our systems. We have fed them with a variety of different things. At the moment, we're feeding with kind of an artificial feedstock so that we have control over exactly what that composition is, but we're trying to use complex molecules rather than glucose or something simple. We have automated pH and temperature measurements. We can take samples that then we can look at volatile fatty acids, which is something that the anaerobic digestion community typically measure, and I'm not really sure why, because it doesn't seem to have any bearing at all on what the community is doing. I mean, clearly the volatile fatty acid levels change, but they don't seem to correlate with anything obvious in the system. But that's one of the typical measurements that, you know, people measure and worry about. We also measure gas flow, both the volume and the composition of the gas. And we've started taking samples where we can extract the nucleic acids and process in more detail for metabolites. Okay, so I'm just going to show you kind of the preliminary data that we have. So on one of the earliest slides, what I showed was we just have set up a GC system so that we can measure small or short-chain volatile fatty acids, which is what the water companies and other companies seem to be interested in, and we can track these over time. So this is a 40-day experiment. That by far the largest by quantity volatile fatty acid is acetic, which is perhaps what you'd expect. And obviously, you would not expect there to be high acetic acid if your process is working well, because the methanogens should be converting it to methane. At the same time as we did that, we took DNA samples every few days. We initially processed a couple of those. We ran them on a small ion torrent system that we have in the department to sequence them, and we generated about 1 billion base pairs of DNA sequence, which we then ran through the MG-RAS server over in the States.
Sorry, and how did you decide which microbes? I mean, because one thing is what you feed into the system, another is what kind of microbes you start with. How did you decide that? So
for what we've seeded the system with is basically from another anaerobic digester. So because it's the wastewater digester from York, we assume that most of the microbes in there are going to be human gut microbes, but obviously you get all sorts of runoff into our water system. So it's going to be a very mixed community. We know that community, because it's coming out of their anaerobic digester, is methanogenic because it's producing a lot of methane. But actually that was one of the things that I found really interesting. So our day 1, day 0 time point is basically what's in that system. And then we sampled again at day 25 to see how our system had changed. Right, so what I've done here, and I'm sorry you can't read any of this, I will redraw it at some point, is look at, so MGRAST basically takes all those short read sequences, it queries them against all known databases, and reports anything that has an e-value of better than 1 to the -5. So they don't have to be good matches in order to be identified as belonging to an organism. And then I've asked it to show us everything that makes up more than 0.5% of the sequences in our data set. So we're not looking at anything that's not very abundant, we're only looking at relatively abundant organisms. So this is what we see at day 0, day 1. And the thing that was surprising to me was that methanogens in the anaerobic digestion system that's working extremely well in York are almost undetectable. They're certainly less than 0.5% of the population in that system. And that may not be surprising because obviously there's a massive influx of microbes the whole time, and most of them won't be methanogens. The other thing that I maybe naively was surprised at is the fact that nearly 45% of the sequences that we get back don't match anything in the databases, even at that low cutoff. And I did complain bitterly to the person that had done the sequencing about the fact that, what is all this stuff?
I think this is always the case, though.
It seems to be. It seems to be. So, and then this is what happened when we looked at day 25. So we see a lot of organisms, particularly things like bacteroides, which I guess are very abundant in the human gut. The numbers of those don't really change very much. But we saw now 10% of the population belong to methanogens, which is quite nice. We see some organisms appear and some organisms disappear. And at least again, at this very crude cutoff, quite a lot of things don't change. So based on these two data points, we tried to identify a few interesting organisms, some that changed, some that didn't, to design PCR probes to, so that we could do some qPCR over the rest of our time course to try and see whether these 2 points were telling us exactly what was happening, whether everything went in a straight line from one place to another or not. And that's shown here. So we've sampled at day 1 and day 25, which is here. I've had to use several different scales for our qPCR. So these are fold changes from the starting point. So you can see at day 25, just as is reflected in our data, the number of methanogens, particularly methanosarcina, have increased nearly 500-fold from their starting point. But interestingly, by day 40, methanosarcina had increased 1,000-fold compared to its starting point. Other organisms, there's a lot more fluctuation or no fluctuation at all, and that was the whole point of this, was to try and see what the dynamics look like. And in fact, now we've managed to find a bit more money. We've taken all these samples and we're going to do next generation sequencing on everything to try and get a better picture of exactly what's going on, rather than just looking at selected species. So I guess our big challenge here is trying to make sense of the data. It's become very easy to generate the data. It's clearly going to be a lot more difficult to make useful sense of it. So going back to that sort of point that there's all that data that doesn't match anything in the database, we also took our sequences and we tried to build contigs from it, because it's a metagenomic data set. It's not targeted 16S sequencing. And so we took those two samples, we pulled all the reads because we assume that anything that's in the second time point was also in the first time point. And we assembled about 31 megabases of contigs from that sequence. Our largest contig was 204 kb, which is a reasonable chunk of DNA from 60,000 reads. The interesting thing about that largest contig is it doesn't come from anything that's in the databases. So, this is a significant proportion of the organisms that are in our mix, because otherwise we wouldn't get that much DNA back from it, and it doesn't match anything in the databases at the moment, probably because it's not something that can grow on its own. And in fact, our 25 largest contigs all came from organisms that aren't in the databases at the moment. Then we got a methanogen, and then we got something called Candidatus cloacomonas acidominovorans. So we've taken, and again, we're very early in the process for this. We've taken our largest contig and tried to isolate open reading frames or identify open reading frames. We managed to find something that looks like an RNA polymerase subunit, which should be ubiquitous. And so we've drawn a tree just based on that RNA polymerase. Obviously, that's not necessarily going to give us a highly informative tree. But this is where it falls when we look at a whole bunch of other archaea. It's an archaeal organism. If we zoom in on it a little bit, we can see that at least based on this gene on its own, and we are in the process of trying to draw better trees, this is a novel organism that has very few organisms that are even similar to it. In fact, in the last year, this organism, Methanogenic, has appeared, and it's closer to that than anything else, but it isn't very close to it. So again, for me, this is very interesting that there are organisms that are clearly quite important for this anaerobic digestion process that we don't even know are there at the moment because of the way things have been studied. So that's really all I have to say at this point. We're actively pursuing ways of measuring these populations. We would like to be able to, take that information, build it into a model, and understand which organisms are important for anaerobic digestion and how those organisms change if they're fed a different substrate. What subset of those organisms are important under all circumstances, if any? What specialist organisms appear if the anaerobic digestion system is challenged with a particular substrate? And that's very relevant, particularly again for these food processing sites, where we've been to a site recently where they're really struggling to get AD to work because of something in their waste stream that the microbes don't like. So being able to identify organisms that can process that and process it effectively would be really interesting. Right, and then, okay, these are the people that I've been collaborating with, students and postdocs that have been in the lab, and people that have provided money for the work. Thank you.